start again by saying that the immune system is a perfect system for applying systems biology approaches to the human immune system. And that's a question of strategy, because a lot of things, we've heard a lot of great work on the mouse model, but there's so many things you can do in the mouse and that you can't do in people. You really need, I think, a different menu of strategies. And so this is one such strategy that we've seized on, and I think has tremendous potential, short-term, long-term. You'll see some of the fruits of that in the work David will present, and he's really been the principal person responsible for this in my laboratory. But just to reiterate, what is a systems approach? Basically, it's when you're looking at any kind of system, a neurological system, endocrinological system, there are a whole bunch of interlocking parts that that hopefully you can measure. Immune system, we can measure these things. There are cell types, there are cytokines. You've heard some of this already, particularly in the various talks, particularly Holden's talk on cytokines. But we have a whole lot of things we can measure. We can measure cells coming and going, and activation states, what cytokines they're making, et cetera. And the systems approach is basically looking at these high throughput measurements with different perturbations. And the perturbations can be drugs, they can be aging, they can be autoimmunity, it can be illness, vaccine, so forth and so on. And you see whether how does the system work in response to these perturbations? So which things go up, which things go down, which things move together, which things move separately, which things don't move at all. And looking at that sort of aggregate, you can hope to discern particular states, particular interactions that you didn't know about before. And I think there are a lot of interactions that we don't know about very well in the immune system or at all that we could potentially discover. And it's somewhat different than the sort of classic molecular biology approach, which could be summarized as, one gene, one postdoc, that you really focus, and look at some piece of something on some cell. And then consequence of that is you don't talk to people that are looking at some other molecule, even on the same cell, because there's really not much to talk about because you're so focused. So I think this is the higher level that, somebody once told me they were sure they were meant to be a biochemist because they like to take things apart but couldn't actually put them back together again. But this is, so what we're trying to do here is we're trying to put the pieces together and see if we can see anything that makes sense. And I think it's a way in, the human model, if you will, is particularly difficult in terms of finding a way in. This is a way in that we can do technically now with ever greater sophistication. Holden gave you a sense of that. And it just applies to everything, basically, or you could try to apply it to everything you might be interested in about the human immune system and health and disease and so forth. So, okay, fine, that's a great concept maybe, but what do you actually do? And what can you do that can get you away from some of the agonies of regulatory approvals and so forth that associate with clinical trials? Well, one thing that was clear is what we could do was vaccines, and this is something, the whole program had already been started with Corey Decker, head of the Vaccine Center at Stanford, and Harry Greenberg and Arvin. We started a whole program to say, well, maybe we can use particularly flu vaccination to understand what's happening, an immune response beyond just the standard antibody tests, which is all that most people ever do. And flu vaccine is particularly good because it's a crappy vaccine. Now, why is that good? What's the evidence it's crappy? Well, elderly people aren't really protected very well, or at any great extent, less than 40% here in this data, but less than 20% in a bad year, like last year. That means you have a lot of non-responders. And that's good for clinical studies because it means you don't have to do thousands and thousands of people. In a yellow fever vaccine, for example, is a really good vaccine. I think 99% of the people respond well to that. So how many thousands you're going to have to do to get a reasonable cohort of non-responders. So flu is very good because there's lots of non-responders. Even healthy young people have a significant number of non-responders. And people are still talking about improving the flu vaccine. There are different things in the works, but it seems to be glacially slow. But anyway, the point of using a vaccine is that it's a safe, if it's an approved vaccine, it's an easy thing to do in terms of regulation. It's safe. Nobody worries about safety issues really particularly, and you can apply it to as many people are willing to volunteer for the studies. Flu vaccine also, most vaccines have some restrictions in terms of age or the cohort that you can use them with. A flu vaccine has almost none of those. Almost everyone can get a flu vaccine, from babies to elderly people, sick people, healthy people, et cetera, et cetera. So it's very broad. And it's also a way of stimulating the immune response. The other good thing about it is there hasn't really been that much work. And so aside from antibody responses, we know very little, or have known until recently, very little about anything else that might be going on in the immune system. The other thing we've done that I think is innovative in the context of ITI and also in the context of flu vaccination is to rethink the whole pipeline or workflow, if you will, of how do you connect the clinical world with the basic science world. This is a big problem because in most cases, the basic science labs are working on mouse models, and they're sort of self-contained universes that might interact within a particular field, a particular molecule of a particular field, but not so much broadly and rarely with the clinical world. And yet here is a particular situation which lends itself exactly to creating a more integrative and cooperative model, where particularly we basically divide up. I mean, you don't want me consenting patients or even bleeding patients. I mean, I think if you trust me, it's not really going to work in your favor. We really need experts. We need people with tremendous expertise, like Corey Decker has decades of experience in vaccinology, has a very good team of people that are expert in giving vaccines and taking blood and consenting patients and recruiting patients and following up, because there's also a follow-up process. There's a clinical database of what meds are they on and so forth and so on. So it really made sense to let Corey be Corey and run totally the clinical side of the vaccine study. And also we add in Holden, and he just told you about his great facility there, which has all these high throughput immune assays. And so basically have a workflow in which patients are recruited, vaccinated, and bled different time points and followed for health issues and so forth, that the samples then get to Holden. A whole lot of assays are done. And then waiting outside Holden's door are a whole cohort of local research people, including myself and lots of great collaborators, who look at this information, trying to interpret that information, accessing samples so that they can do different assays, novel assays, and so forth, and interpretations. And we have a whole gamut from T-cell people like myself and Chen, B-cell people like Harry and Jean Butcher, Xiaosung He, PJ Utz. Jörg Granchi is an expert on aging in the immune system response. Scott Boyd and Andy Fire working, as well as Steve Quake, on antibody repertoire analysis. So there's really fantastic new technology for sequencing millions of antibody or T-cell receptor sequences, looking at that data, looking at those responses. Those are very complicated responses, but the technology is here to really encompass a good chunk of that and to learn just what is happening in different people and in cohorts of people in terms of their repertoire responses. We also need really good computational people because, again, you're dealing with large data sets. You need skill, you need new analysis methods in terms of computation. We've gotten that. I don't have Professor Katrian here. You'll hear a little bit more about his work with David, but he's got great stuff there. But we've also worked with Daphne Kohler, Atul Butte, Raptib Sharani, who are some of the preeminent computational and statistical people on campus. We also, I think there's a crying need for new technologies to look at human samples. There really hasn't been I think, a very aggressive development of the kind of technologies you need to analyze a human blood sample. Most people are happy to get a blood sample. They do analysis of a dozen or so different parameters. With the technology we have in the immune monitoring core, plus some of the other technologies, we're easily looking at 50,000 variables in a blood sample. So a lot more data, that's good, bigger net to cast, look at all sorts of aspects of the immune response. But it's also, of course, and Holden touched on this too, is the need to be able to manipulate this data and analyze it and ask it questions and get reliable answers. But also we've done some great technology development work with Ron Davis, Michael Mandrinos, my own group, PJ Utz, Bill Robinson, and others here. And just lastly, what have we got to show for this after now about 6 years of effort or so? I think it's pretty good, and you'll hear more specifics from David, but in terms of particular studies related to sex. But we basically have, I think, over 500 healthy subjects have gone through Corey's center, been vaccinated, had blood drawn, had that blood analyzed at the immune monitoring core facility that Holden talked about. This includes a longitudinal cohort, which is very unique. There are almost no longitudinal cohorts in immunology, and yet, as question that Sabra asked, there's a huge need, because you really, what's predictive, what's not predictive? All these diseases like cardiovascular disease and cancer and so forth have this immune component, but to what extent do you see something going on in the immune system before these events? And so the idea, this longitudinal cohort, 60 of the 90 individuals are 60 to 90 years old, 90 plus now, and we've been following them, and that's, we have all this data from years and years before. In the cases of several of them have died. We predict that more of them will die, and we hopefully can capture some connection between something we might see early on in the immune system versus later on. Also, we've, with Gary Swan, who has the largest twin registry in the Bay Area, about 3,000 twins now, we've been working hard to pull a good chunk of those twins into the Corey's Vaccine Center and analyze them with respect to vaccine responses and all these other immune parameters. No one really knows that there's been a little bit of work on specific immune parameters like certain cytokines, certain cell types, and whether they're genetically driven or not. But no one's done a really comprehensive study. And bottom line from our study, which has been headed by Peter Broden, is that looking at 212 twins ranging in age from 8 to 80 years old, is that we find many of the immune parameters, the variation in the immune parameters, is driven by environmental influences, not genetic influences. So, and that makes perfect sense because the immune system is a sensory system. You only survive if you can adapt to your particular environment. Your hearing, your other classical neurological senses, but it's hard to think of something more important than your immunological sense in terms of, whether you're able to survive in a particular environment or not. So this makes sense, but it's different than what people have seen previously from largely fragmentary data. And as I mentioned, also we have made large investments in immune monitoring technology. I think we now have more site-off machines than the entire East Coast of the United States, just to throw out a number. We have can do large arrays of peptide MHC tetramers, over 100 different tetramers at one time, to analyze T-cell responses. We developed the low-cost HLA sequencing technology that's going to be really helpful in understanding the relationship of HLA type with all sorts of immune phenomena. So I will stop there and turn it over to David. Hopefully you can find his presentation here on the machine.
Thank you, Mark, for this great introduction into how to look at this data and how important systems biology is for human immunology. I've been working with Mark in Mark's lab using systems immunology approaches to understand sex differences in the response to influenza vaccines. And well, we well know now that women have a stronger immune response. That can account for their longer lifespan. They have more responses to vaccination infection. And this might be explained by two theories. One was mentioned before, the immunocompetence handicap hypothesis that is based on the dual role of testosterone, and also the Bateman's principle that is more of an ecological explanation. And it says that males invest more energy in competing with other males, but that has a trade-off for the immune system, whereas females maximize their fitness via longevity, so they invest more energy in keeping a healthy immune system, at least in premenopause women. So this will be a diagram how the Bateman's principle will work. So males will compete for a limiting resource, which is females, to reach reproduction, because they have, on average, less chances of reproduction. And this is just to illustrate that males have about tenfold the amount of testosterone that females have, and also the amount of estrogen and progesterone during females' lifespan changed quite dramatically. So as Mark mentioned, we're using this cohort of 90 individuals, young, older, CMV positive and negative, females and males. And they come to the clinic, we get blood from them before vaccination with the influenza, inactivated influenza vaccine. And we come back 28 days later, and we get another blood draw to study the antibody responses to that particular vaccine. So we collect a large number of parameters, including whole blood gene expression. We generate gene modules, serum cytokines, some chemokines from serum, from blood, and cell substrate frequencies, up to 19 in this case. And we analyze cell function by looking at phosphorylated status of STAT proteins in many different cell populations. So a total of 278 parameters. We analyzed differences between males and females, and we found differences only in 7. And most of these have been described in the literature. So leptin, as was mentioned by Holden, when arrays, so particularly inflammatory markers. And we found, so in females will be in yellow here, and males in blue. And so there was only one given feature that was increased in males, and this was a gene module composed of Y chromosome linked genes, so not surprising. We also observed differences in the neutralizing antibody response to flu, with a pretty strong regression coefficient, minus 0.7 for H3N2 strain and minus 0.6 for the B strain. We observed no differences for the H1N1. And this is consistent also with the literature. We still don't know the basis of this. So there's definitely a sex effect on vaccine response, and we wonder whether there's a feature that might be intermediate between this, the effect, and that could explain differences. So in collaboration with Rob Tipriani here at Stanford, we search for confounding factors, which is important when you do feature selection. And we did that by stepwise regression. So the way it works is illustrated in this function here. So on the left hand side, there's the response to vaccination that will be equal to sex times the regression coefficient of sex plus feature x times the regression coefficient of that given feature plus a constant here. And we add the 278 features one at a time and observe changes in this regression coefficient of sex of more than 20%, and those are included as a confounders. So we observed 2 confounding factors. One is module 42, that is composed of ribosomal proteins, and the acute phase inflammatory protein CRP. So we included these 2 features for the further analysis. So then we searched for significant interactions with sex that could explain vaccine response, and we fit the model to estimate odds ratio. We found only one feature that was significantly associated with sex and could explain vaccine response, and this is module 52, which is composed of 35 genes participating in lipid biosynthesis. And also some interesting genes here were LTA4H and PDSS2, because they participate in the development of suppressive cells from the myeloid and from the T cell compartment as well, Tregs specifically. So as you can see here, per unit of module 52, the odds ratio of being a responder in females was 2.2, whereas males have a very low probability of response. Something to mention, I'm going to touch on this later on, is that the regulatory program of this gene module contains several genes, including FOS and JUN. So the AP1 complex as being a repressor of this of expression module 52. And those have been also shown to interact with testosterone in previous studies. So given this very, very important different effect of module 52 in males and females, we search for gen chemical associations using databases from the public domain for association for testosterone, estrogen, and progesterone. And we only found significant association for testosterone. So in other words, there were previous studies where they stimulated with testosterone different type of cells, and they observed that there was down or up regulation of the genes in this particular module. So we went ahead and measured testosterone levels in the individuals from our study. And we divided the males individuals into testosterone high or testosterone low if they were above or below the group median. And as you can see here, the lowest response to the vaccine was observed in those males with high levels of testosterone, and the significance for the interaction with sex was only found for this group, but not for males with low levels of testosterone that seem to respond pretty well, and expression module 52 does not do much to the responses to the vaccine. So we generated this little model here where testosterone might inhibit the AP1 complex, which in turn shuts down expression module 52, participating in immunosuppression. So if you would have high levels of testosterone, AP1 will be downregulated and there's immunosuppression. Of course, this remains to be tested. We can go ahead and test this hypothesis in the laboratory now. I would like to mention now a new study what we're doing, particularly with the collaboration with Dr. Khatri, using data from the public domain. So in essence, what he's been doing is pulling up a lot of different studies, dozens of studies, hundreds of samples, and trying to get signatures that can distinguish between responders and non-responders to flu. This data has been deposited in public repositories, so you can use it freely. And he found a signature of 11 genes that can very nicely distinguish between responders and non-responders to flu. And we asked whether kinetics of this gene expression is different between males and females. And as you can see here, the expression of this gene signature at day 3 was higher in females, so essentially picks up day 3 and remain constant. And this is statistically significant. Whereas in males, this signature will peak at day 1, and then it goes down and returns to baseline. So how is this possible? How is that in males, expression of these genes that went up day 1 after immunization, then goes back to baseline. There's this study that was done a few years ago. Actually, is a meta-analysis of a series of studies published where they either activate the immune system or immunize or infect the immune system of different species. and they measure the levels of testosterone after immune activation. And as you can see here, these are the different studies. The effect size is always negative, so testosterone drops dramatically after immune activation. And so this could be one of the mechanisms by which males are dropping their gene expression of these particular signatures. But again, this generates a large number of hypotheses that then we can go ahead and test it in the lab. So to conclude, testosterone seems to be pretty important in the regulation of the immune response to vaccines, in particular to influenza vaccines, maybe others. AP1 could be implicated in the mechanisms. And gene expression data from the public domain can be used to study vaccine-induced differential expression. There's different kinetics in males versus females, and testosterone could be implicated in these differences. So with that, I'd like to thank people from Mark's lab that have collaborated with a lot of different assays in analysis and discussions. Katri lab, people from the University of Bordeaux. This was a collaborative project. The clinical core at Stanford, HMC, Rob Tufsharani, Daphne, and funding. Thank you. Questions? Yes,
in
Corey.
the males where you saw the association of increased testosterone and low responder status, did you do an endocrinological workup on those individuals to see whether there might be another abnormality which is responsible, or did you look at androgen receptor activity because there's a feedback inhibition mechanism in which if you have a defective androgen receptor, you'll have higher testosterone. So it might not in fact be just testosterone in and of itself, but it could be just regulation of endocrine activity in males as a consequence of some other factor.
Right. We haven't looked at androgen receptor. I've been studying androgen receptor, both the nuclear androgen receptor, the classical one, and the non-genomic pathway in different cell types, in immune cell types. And this, we're planning to do a much larger and broader panel to study androgen receptor and estrogen receptor, different estrogen receptors and progesterone receptor in different immune cells using the CyTOF instrument. But this is something we're planning to do. We haven't done.
You have to make sure you include the neuroendocrine pathway in terms of regulation of testosterone production, because there could be something coming from the neuroendocrine aspect that's regulating testosterone production as a readout that could also influence adrenal function, for example, or some other mechanism.
Right. Well, that's true. Thanks. But I just wanted to mention that one possible mechanism of this drop, at least what we see here, is IL-1. IL-1 has been shown to shut down Leydig cells from producing testosterone. So that's something we should definitely pay attention to. Thanks. Yeah. Other questions? No? Thanks.